With this video, what I want to really focus in on is how the three major macromolecules that we've looked at are involved in the cell membrane. So what is the role of lipids in the cell membrane? What is the role of carbohydrates in the cell membrane? And then lastly, what is the role of proteins in the cell membrane? And I actually have a whole separate video that looks at the function of transport proteins. And in the context of talking about lipids, we'll get into the discussion about how it is that molecular properties of a substance or a molecule are going to influence how it enters the cell. And those are going to be some of the key takeaways from this video here. So with the plasma membrane, right, it truly is a mosaic. And when you think about a mosaic, you think about all these beautiful colored tiles that are compiled together to make art. The model really fits with the cell membrane. You've got carbohydrates coming off with glycoproteins and glycolipids. You've got your phospholipid bilayer. You've got all of these proteins making up channels that allow for entry and exit across the cell. And you also have these cholesterol molecules that are going to be embedded within this phospholipid bilayer to help with fluidity. And this is all moving. It's highly dynamic. And so this is a great way to think about what it is that this plasma membrane really looks like. It's very dynamic, constantly responding to the environment. So when you think about the environment that our cells live in, I want you to bear in mind the fluids that surround our cells and what's in those fluids. So to look at the composition of water in the human body, you have water in our bloodstream. So that's going to be our plasma or our intravascular fluid, intra meaning within, and vascular, our vascular system. You have the intracellular fluid, so again, within the cell. And then you have the interstitial fluid. And so the interstitial fluid is neither in our bloodstream nor in cells. It's part of what we consider to be the broader extracellular fluid, but it makes up a lot more of our overall body composition, our overall breakdown of water percentage in the body than many of us are aware of. And so I love this graphic because you're seeing, about 60% of body weight is made up of water. We'll say 40 liters in the average human person. Of that, 25 liters is contained within the cells, right? That oftentimes surprises people. You have about 3 to 5 liters, we'll say, that's going to be in your blood. And then the interstitial fluid, it's about 12 liters of fluid in your body that is going to be in this space. And within that, you've got amino acids, you've got fatty acids, you've got sugars, you've got vitamins, you've got hormones, you've got cellular waste, you have lots of different salts and ions. And so I'm trying to frame for you a picture of what our cells are swimming in, right? It's this incredibly dynamic environment, and they have to be very selective about what they pull in. And so I like to think about our cells as being waterproof, right? And we'll look at how they make themselves waterproof, but they are fairly impermeable to polar substances. And just like a cell phone that you would want to be waterproof, right? If this were permeable to water, it would damage the phone. It would damage our cells if it were not selectively permeable. So if the cell were capable of just freely bringing in anything that were in this interstitial fluid, it would be damaging to the cell. So this idea of selective permeability is critical to the health of our cells and the health of our body. So let's look at exactly what this phospholipid bilayer looks like in our cells. I know you all have seen this content before, but it's worth repeating because it really is critical. So here's just your typical cell. And I emphasize here that our cells, right, are swimming in a watery environment. Our bloodstream is made up of water. All that interstitial fluid is made up of water. And so our cells are very much swimming in a watery environment. Similarly, the cytoplasm inside the cell is also going to be water. And so what we see is that our phospholipids, right, here is your phospholipid in isolation. So again, you've got a hydrophilic head. So hydrophilic is the same thing as polar. It's also the same thing as non-lipid soluble. So I have this vocabulary highlighted for you all because it is important to be able to keep these terms straight and to be able to use them interchangeably. You'll see different images refer to different names. Oftentimes you see this as a polar head, but it is also water-loving, hydrophilic. The tails are water-fearing, hydrophobic. And you can also describe those tails as being nonpolar or being lipid soluble. And so these phospholipids, it's a bilayer because they are arranged in a layer of two. And so you have them oriented where the polar heads, the piece that's water friendly, is going to be exposed to both the interstitial fluid, right, the fluid outside of the cell, and in addition, the other layer as phospholipids is going to be oriented where those polar heads are facing the inside watery environment of the cell as well. So it's important to appreciate the orientation of these phospholipids and why they're oriented in this way. And so one of the key takeaways for this video is to be able to appreciate how polarity of a molecule will influence how it enters a cell. So we have our phospholipid bilayer shown here. And what we see is that any molecules that are lipid soluble or non-polar, they can diffuse right through that cell membrane. They don't need to have any special doors, any special transport proteins to gain access to the cell. They are not going to be repelled by the lipid tails embedded in our phospholipid sandwich here, right? So they can just go right through the membrane. On the flip side, anything that is non-lipid soluble, anything that is polar in nature is going to be repelled by the fatty middle, by the lipid soluble center of our cell membrane. So they are not going to be able to gain entry into the cell unless they have a special port, unless they have a special door. And these doors are going to be proteins. So you see a membrane channel here, and if that membrane channel is specific to a certain non-lipid soluble molecule, then that specific molecule can gain entry. Now, another thing that's highlighted here is that ions, so these charged molecules, you'll see will also need to have a membrane channel in order to gain access to the inside of the cell. So these are going to be calcium and magnesium and sodium and chloride. All of those ions carrying either a positive charge, are cations, or a negative charge, are anions, are going to need a membrane channel. To summarize what I've already said, so one of the key factors that determines if a substance can diffuse across is its lipid solubility. And the other thing that I haven't yet addressed is simply the size. So water is one of those substances that is polar in nature, but it's small enough that it can diffuse through the membrane. You'll actually see that what it's going through are what's called aquaporins, so special pores in these membranes. Certainly a large molecule like glucose, which is both large as well as polar in nature, it will not be able to gain entry. It will not be able to plow through this membrane unless it finds its special transport protein. Amino acids, the same, too large, usually too polar. Oxygen, CO2. So those small gaseous molecules, they are small enough that they can diffuse through the membrane. These are also nonpolar, so they have two factors working in their favor to allow entry into the cell. So turning now to the role of carbohydrates in the cell membrane, and what I really want to focus in on here is what's called the glycocalyx. And so you can very much think about this as a sugar covering of a cell's surface. So here's a depiction here of what this glycocalyx looks like. And the glycocalyx, so you have some carbohydrates that are going to be attached to lipids. You see them attached to cholesterol here. You also have other carbohydrates that are going to be attached to proteins. So those are simply termed glycoproteins. But what you end up, all of these green tags on the surface here, that is showing you how rich the coating is of sugars on our cell surface. And one of the functions of this glycocalyx is to help with cell-to-cell recognition. Every cell type will have a different marker, will have a different glycocalyx on its surface. And so from an immune standpoint, this allows cells of our own body to distinguish to distinguish a cell rather that is from our own body self from non-self, potentially a foreign invader that could cause harm in the body. I wanted to include two nice examples here from the human body. Take, for example, the sperm cell. We see that embedded within this glycocalyx are certain cell adhesion molecules that guide these cells, that allow them to adhere to other cells. So when the sperm is trying to bind to the egg, the glycocalyx, which is everything you're seeing coming off this graphic. I realize it's grainy, but it's just showing you different glycolipids, glycoproteins that are coming off this aspect of the sperm, the head of the sperm. And so again, the glycocalyx in this case helps to really guide cell communication, cell adhesion. We also find this really rich glycocalyx that lines the endothelium of our blood vessels. So lastly, the role of proteins in the cell membrane. So about 50% of your typical cell membrane is comprised of proteins. So it is just jam-packed with proteins. And I think that makes sense when you consider how many things rely on those proteins to gain entry into the cell. So again, I have another video that's focused on this, but you have passive transport, which does not rely on ATP. And so what we're seeing here is that with passive transport, we are always going down a concentration gradient. So we are going from a situation of molecules that are highly concentrated to going down that gradient to a place where those molecules are far less concentrated. So high concentration to low concentration, that's always going to be the case with passive transport, no matter whether you're talking about diffusion or osmosis, the case where water is moving down its gradient, or facilitated diffusion. Just because you have protein transporters doesn't mean that we are not still bound by this principle of going down a concentration gradient. With active transport, we can move molecules independently of that concentration gradient, meaning that we can move molecules from a low concentration to a high concentration depending on cellular needs. And that's a situation where you would depend on ATP to cause that transport. And so hopefully at this point, you feel as though you could highlight some of the key properties that influence cell entry. So the polarity, as well as the size of a molecule. And you could describe how the role of fats influence the cell membrane, so our phospholipid bilayer, the role of carbohydrates, emphasizing the glycocalyx, and then the role of proteins and their importance in influencing membrane transport.